So really wanted to think about, the fundamentals here, sort of like what is lab automation? And we often look at sort of the work that we do in the lab, and we think about ways that we can improve our efficiency, we can reduce errors, increase accuracy and throughput. And really the only answer for that, at scale is automation. But applying it is challenging, and we really need an understanding and a strategy on how to do that and how to go about automation in the lab. So we'll look at sort of this progression, starting out manual pipetting on the bench, using standalone instrumentation, but then moving towards benchtop automation to increase throughput, and then building out sort of what we call work cell automation, and then factory level automation. The key then is to find the right place for your team at the right time, and how to build out your capabilities to match your needs without taking on too much and without falling behind the needs of your lab. So traditionally the first step is this benchtop level automation. It's still focused on people moving samples around, but gives you the opportunity to gain a little bit of additional walk-away time, to have the liquid handling managed by a robotic system rather than a scientist. Once that sort of reaches its sort of peak efficiency, you would move to more of a work cell level automation. And this is really about taking instrumentation really off the bench and dedicating it to a position within a work cell that is usually centered around a robotic arm that can move the samples around. So again, you're reducing the dependency on human beings here within the system and allowing a robot to take over some of the more mundane aspects of it. And so again, though, this is targeted at usually assay level work and trying to solve a particular problem, within the lab, one or more assays that you can sort of dedicate this hardware to. And the sort of later stage within that system is this more production level, fully automated and orchestrated systems that you would see. And this differs in the lab quite a bit from the way it is applied in industry. And Imad will talk a little bit more about that later, in that it is usually built around sort of more dynamic workflows, especially in the R&D space. There's a lot of evolution that happens in your assays over time, and you're not tooling up to run the same process for 5 years necessarily. So why would you apply automation? What are the real benefits of this? The key here is walk-away time, and it's freeing scientists up to do knowledge work rather than sitting there, again, pipetting or waiting for samples to be completed to move to the next step. And so really, you can free people up, but then you can also extend your operational time up to 24/7 operations within your lab. Another key pillar and sort of reason to automate is throughput and scale. And this is a really interesting one because you're both often needing to scale up your production, but also scale down your size. And so you can achieve tremendous savings on reagents and samples by miniaturizing your assays. And after the great discussion of microfluidics, there's really this whole range where you can go as you're starting to miniaturize your assays. And the challenges there become simply that human beings aren't even capable of pipetting at those volumes, right? And so there's a whole range of automated technologies that can be applied that can give you that kind of throughput and that scale. And so again, scaling down in size and up in throughput. Reproduction and traceability, or reproducibility and traceability are two other keys here. Everybody hears a lot about the reproducibility problem in science, right? Is how do you really know what happened? And the more that you can remove the human variability in things, the more that you can get consistent data and be able to reproduce your experiments. And if you can really clearly define how it's done, you'll have a lot better chance of reproducing it in the future. Error reduction and data integrity. As you mentioned in the intro, data is critical, but the common process in the lab, right, is to take notes in a notepad and then a week later or later transcribe those into, even if you have a digital electronic notebook, it's often not how things are actually captured directly. And so again, if you can remove that human element, you can get real traceability on what happened. how long did those samples actually sit on the bench? What was the real incubation time? And then again, data integrity. How many times do you see people moving Excel files around on flash drives, copying things out or pulling the wrong lists, because of that, human in the loop concern. So again, as you're looking at these things, these are all keys. And finally, physical safety, right? There's repetitive stress disorders that you can get from working on these very repetitive jobs. And again, the use of hazardous material and being able to really separate people from that. So as we think about automation and as you apply it to your lab, I want to think a little bit more nuanced about this though, because people often think about, let's just automate, let's throw some robots at this problem. But the real key here is, I think, the difference between automation and integration and how they play together. So automation is really trying to reduce the reliance on human assistance within your thing. So you can have a robot moving samples instead of people. But integration is how do you connect systems together to ensure the seamless transport of data and samples. And what I've always worked with people on is how do you apply sort of an integration first automation strategy, where even if you have people in the loop moving samples, you want it to be as integrated as possible so that the data is transferred seamlessly, that there's tracking and traceability on your samples. And so with that focus, you can then scale to fully autonomous or automated systems much easier if you focus first on the integration aspect rather than on the physical automation of a platform. This leads into sort of a different way of thinking about automation that has become sort of a really key driver in the industry right now, and that's this concept of orchestration. And it's not always just about, again, sort of robots and automation. It's how do you seamlessly and intelligently coordinate everything that's happening in your lab. And again, whether you have people moving things or robots moving things, you can do a lot to improve the performance of your lab and provide, that sort of the guarantee that a lot of the benefits that we talked about of automation can be provided through this concept of orchestration. And it's really simply this, is that through the processes in your lab, you have samples flowing, you have data being passed from step to step, inputs and structures of one experiment, outputs throughout, and then you have these consumables, reagents, and wastes that are managed throughout this whole process. And again, whether they're manual, whether they're desktop, benchtop automation, or whether they're work cell or even full automated platforms, orchestration is how you manage the flow of all of those through your system. Sort of 4 key foundations here: workflow management, your liquid handler methods, how your readers are configured, all of that needs to be managed and thought through, and so that you can again move anywhere you want to within that sort of spectrum, that continuation of where you're automating. But you need to start thinking about workflow management. You need to think about robotic automation, and then material handling. Where are your samples coming from? Where's your labware stored? What do you have in inventory? Everything through that, and then data capture and analysis. And by sort of looking to manage all of those, you can really provide sort of an orchestrated overview of your lab. Key challenges here, of course, nothing's ever as easy as it should be. Scientific and engineering challenges. Biology is fuzzy. There's often scenarios where there's variability in materials, samples, processes. It's often difficult to quantify exactly what you're doing. There's a lack of standards in the industry, and that's a huge area that really needs to be solved. And then there's just the other, just general engineering challenges, capturing all of that data, managing that throughput, staffing. And so then, that leads to resource limitations, right? How do you, afford the cost of the instruments? What's the ROI on that investment? And how do you ensure that your team has the right skill set that they can really manage the automation that you bring into your lab? Again, it's a much bigger problem than just finding the right technology to solve the problem. You have to understand the context in which it's going to be applied. And then really the human aspect, the psychology of bringing automation into a lab is something that's really often overlooked. What is the impact going to be on the scientist? Is your lab manager solving problems that aren't the real problem? And so really being able to understand, the significance of the individuals that are doing the work and where they see the real challenges compared to what the high level view of that is. And so bringing in automation, you have to deal with people's concerns about learning a new technology. How do you approach this and really show them the value that it brings to them when they become an expert in this? Because you think often about robots are taking away jobs, but if you can think about it in terms of the value that you provide to a scientist who can now not only do the science, but also run the automation, they become much more valuable to their company than they were without that. So it's key to have an automation strategy. Most people tend to apply sort of a whack-a-mole strategy. They see a problem, they try to fix it with automation, and they go on to the next problem, and they don't take a holistic view of what needs to happen in order to achieve their goals, right? So we have to, you have to think about what you're doing. Again, you might be faced with a problem of throughput. I need to process more plates every day than I can do right now. But you can't think of that as an isolated problem. You have to think about the upstream and the downstream processes. What happens all of a sudden when you need to standardize on a piece of labware for an automated process? How does that impact the team that's supplying you samples and the team that's taking those samples downstream of you? So think about it in context. Also, you need to have automation that works with humans. And I talked about this a little earlier, but really you want to choose technologies that will expand and grow the capabilities of your team. And do that without stretching them beyond what they're really able to take in, what they have the time to learn, everything. Sometimes you're better off choosing a less fully automated system if it's more approachable and easier to use, right? And you think of this as an evolutionary approach to automation rather than I'm just gonna buy the fastest, greatest, best solution right now. So really take into account the team that you have, how you can grow them into the new responsibilities that'll come out of this. And then, again, I talked earlier about understanding the impact on their real problems. And take this sort of crawl, walk, run approach. Don't bite off everything at once. This really, and the application of automation in a lab is a change management problem, and change management is hard. And so, approaching it the right way, bringing people in, building, the confidence, so take small steps, but plan out the future. Don't take the isolated individual steps that'll just solve your problem right now. So with that as sort of an overview to automation, I'll hand it over to Imad, who's online.
I am. Can you hear me? Hi, everybody. This is Imad Mansour. Thank you for being here and giving us the opportunity to speak. I'd like to begin a little bit by talking about what Hover Labs' vision is for automation in life science. We're taking the approach of looking at industrial automation specifically from the perspective of how manufacturing has been revolutionized by specific principles such as the assembly line concept and the use of robots for conducting complex tasks. We feel that by adopting and adapting these proven concepts, lab automation can achieve new levels of efficiency and functionality. The assembly line concept has been massively successful because no time is wasted between each step of a process. They are highly efficient. They allow production processes to run continuously. And their modular nature allows for relatively easy reconfiguration. Also, in industrial settings, robots are mainly used to perform critical workflow tasks such as welding, painting, assembling, packaging, and so on. In contrast, lab automation limits robots to simple transport tasks, moving samples from one scientific device to another, effectively making the robot arm the assembly line. But there are now far better ways to move samples around in the lab. Next slide. Planar robots are flat surfaces with one or more shuttles that move quickly and precisely using electromagnetic levitation. These robots are made up of square tiles that can be arranged in any size or shape. They offer all of the key features of an assembly line with some added benefits, such as massive parallelism, Because configuration options are unlimited, we can run many different processes simultaneously. Unlimited scalability and configuration options, customers, because of that, customers can expand and upgrade their systems without a complete redesign. And because no gripper is involved, sample transport is very reliable. But the challenge and the opportunity is that most liquid handlers and scientific devices can't be directly integrated with a planar robot. An arm would still be required to move samples in and out of such devices, effectively moving the samples of the assembly line off the assembly line at each step of the process. So we have to rethink how liquid handling is done and how scientific devices are accessed. By redesigning pipetters, reagent dispensers, and plate planar robots, samples can drive right up to them without the need of an external arm. Next slide, please. What you see here is a Hover Labs concept for a system that can perform most ELISA or ligand binding type assays. The pipetter, washer, and dispenser are integrated directly with the planar robot. Samples can drive right up to them while moving simultaneously. But many other types of devices will still need to be accessed. And on the far right, you see a simplified, low-cost gripper for doing just that. It allows us to integrate readers, centrifuges, and other types of analytical devices. A system like this is a common entry point for labs automating the processes. As their needs evolve, however, the Hover Lab system can evolve with them. Click, please. This approach represents a paradigm shift in lab automation, one that embraces the concepts of industrial automation while meeting the complex needs of the lab in new and more interesting ways. It releases us from a lot of the constraints and limitations that are associated with traditional lab automation design scenarios. Next slide, please. So what are the key benefits? As I just stated, it does give us a new way of automating scientific processes, one that can open up entirely new approaches to automating our science. It offers us easy configurability for future-proof design. This approach is inherently more reliable and offers greater speed. Time between steps is significantly reduced, leading to greater efficiency. And overall, we believe it's going to be and is a far more cost-effective solution. And with that, I leave it to Steve for the lab robot section.
All right, so I'm going to talk about autonomous mobile robots, which are robots that navigate within the human space to work alongside people to automate tasks without committing resources and infrastructure to full-time automation. Now, these are essentially vehicles that drive around the environment, localizing themselves and path planning to get to their destination and automatically avoid obstacles. Now, they were first released back in 1950s, but not actually realized in manufacturing and logistics space until the late 90s. And they haven't made it over to the lab automation space until mid-2010s. You might have actually seen them in some restaurants or hotels delivering food or towels to your room in a hotel room, but they're vastly utilized in the logistics and warehouse space, shuttling essentially pallets or large boxes around instead of requiring a fleet of forklift drivers. And then moving into the lab automation space, they can be utilized beyond just sample transport, primarily in two main ways. They can use to move samples between different work cell to work cell. In contrast to the previous description that Imad had put together, utilizing track-based systems, these are much more flexible as they can simply drive around in an environment without major overhaul of infrastructure. So they don't need a rigid track-based system connecting all your work cells together or your labs together. Essentially, it can traverse space just as a human can. So going up between floors on an elevator, down a hallway, populated with people, and it can move parts or samples between those different work cells. To really utilize the technology, however, isn't so much in that sample transport. It's actually working directly alongside humans in an already developed lab space. That requires the robot to actually access and control instrumentation directly, but then you could simply have the robot perform the workflow that a technician might be performing in a day-to-day operation. The technician could also rely on that AMR to essentially create a on-the-fly workflow to assist them in performing operations such as retrieving samples from storage or prepping certain labware. Lab robots were advancing those AMRs beyond sample transport. There's a lot of requirements, as discussed, to really take it beyond just moving sample from A to B. It requires an intelligent awareness of all resources in the lab. So it's tracking the utilization and status of all instruments, all equipment within that lab. It needs to be able to access and control that equipment and keep track of all consumables, not just the sample plates that are being operated on the equipment, but also tip boxes or reagents, whatever, to make sure that it can perform the workflow just as a technician can do. We can also leverage new AI tools to make this robot really into a true laboratory assistant. By leveraging generative AI, we can automatically generate the workflow that AMR needs to achieve based on conversation-based queries or simple setup programming, instead of requiring an engineer to think ahead and plan every single workflow and test it in advance. We can also expand that further by allowing the AMRs to collaborate together. Instead of having simply a robot arm on top of the AMR, you could put another instrument or an incubator or something, and then have the AMRs come together to form a self-assembling work cell, essentially creating an extremely modular lab environment without the reliance on a static robot arm and pulling all equipment to one location. It can be changed day to day autonomously using robotics.
So yeah, as you can see, there's a tremendous amount of improvement that can happen to lab automation, and the work that we're doing at Hover Labs and Lab Robots are just two of the ways that we can sort of transform the current static paradigms that I was talking about into much more flexible automation solutions. But I wanted to kind of, in summary with all of this, take kind of a step back. And as I talked about sort of your automation strategy and how you approach automation, there's often this real tendency to dive right in. And it's I've been in lab automation for a long time now and built a lot of systems, and I find myself reflecting sometimes how often I tell people don't automate yet, right? Don't jump in and follow a technology because it's exciting, right? Peter Drucker said mission defines strategy and strategy defines structure. And when you apply that to your automation strategy, look back at what your vision is. There's been too many times I've seen companies that have lost sight of that have gone all in automation, and the science never worked. They had problems in scaling because they were just chasing this disease, this technology without the real why behind the company. So don't lose sight on what you're doing. Your mission isn't to automate. Automation is a tool that can help fulfill your mission, and finding that balance again is really difficult when you got a lot of really excited people about some really cool robots. Carefully evaluate the buy and build decision is another one I see people kind of getting caught up into. And I'll give you a hint. Buying is usually always cheaper. It never seems like that when you're first penciling things out, but it's often a lot more difficult than you anticipated. And that's where you have to really weigh your benefits of time to market and discovery versus chasing technology and chasing ideas, right? So really thinking about those things very carefully. Look at the ROI on what you're doing, and really, really do the math when it comes to that. And the other thing is look for a vendor, or don't look for a vendor. You want to find a partner. Automation is a journey. It's going to take a long time. It's an evolution that will happen in your lab as your needs change. And it's not the kind of, you want someone who's there to help you with your full journey. And so as you're looking at automation, partners, really evaluate them on how they can help you evolve, and someone who's there to help you with your full journey. And so as you're looking at automation, partners, really evaluate them on how they can help you evolve and grow your systems to meet your ever changing and evolving needs. So thank you very much. Thank you very much for your time. Any questions?
Okay.
Oh, okay.
How long does it take to build up
Yeah,
a production line? That can vary a lot, especially with essentially the lead time of buying the equipment. Generally when we're building like a work cell level, if you were one or more robotic arms and let's say a dozen instruments, lead times could be up to sort of 12 to 16 weeks just to get the equipment in-house. And then there's a matter of designing all the methods. And so it's usually 6 to 9 months in the end for the development of a large system like that. But on the flip side, you can, if you already have the equipment in hand, you can bring a robot in and you can get something going in a matter of days. So there's, it's very different between, designing a lab in a big pharma versus let's get some equipment off eBay and, get running really quickly with, robot loading samples to run 24/7 on a, some analysis machine. So it can go very quick, but generally think of it more like a construction project where there's, lead times and a lot of, management of, when you're moving to the lab, getting air and power hookups that are appropriate for the instrumentation stuff. So it can, lab, it's a lot like a lab build out, and then just make it more complex because you've added robots and then software integration and all of that.
Can you hear me okay? So what area of the lab automation do you see expanding the most? Would it be like assay biology? Would it be compound management? What are you seeing at this time?
There's two real areas. So automation really grew its foundation in high throughput screening. But those same instruments, the liquid handlers, the readers, the robots that are used there have proliferated into automating really every vertical within life science. But the two areas that I think the most growth, over the last, say, 5 years, it's really been NGS. But I think the future is in, cell and tissue culture and chemistry. And those are the two areas that we have, tremendous demand, especially with the need. I mean, there are so many more targets being discovered now and so many more opportunities, and automation's really the only way to do that. And so that's where I think there's a rising tide across all of life science for automation. But cell and gene therapy is going to need a lot of cells grown, and that is a time-consuming process on the bench, and one that is automatable, but it's not out of the box easy to do yet, as in the way that a lot of the high throughput screening type automation is. There's managing temperature controlled reagents, and you're running assays over the course of sometimes months rather than just days, right? You're not just screening for the same thing over and over again. And then again, same thing with chemistry. There's a lot of the manual processes that are weighing out solids, things like that are actually challenging still from robotics. But the technology's here, and it's going to have quite a bit of growth in that area.
Thank you for mentioning, the psychological aspect of this. That is a huge deal, and something that immediately came to my mind right as you were talking about that. One of the other things is you guys mentioned, not necessarily eliminating LIMS system, but automating this into an electronic notebook. Now, the question I have about this is, do you intend to offer sort of like a package deal where you have your own software system as well? Or is this simply going to integrate? Because I know that's going to be a challenge too, like perception of like the administrative as well as like learning curve.
The informatics space is a really challenging space right now because it's such a fractured environment. I mean, I talked to a lot of like large pharma companies, and after all the acquisitions and all the in-house stuff, they'll have 20 or 30 LIMS systems or ELNs and systems of record that all need to work together, and they can't get rid of any of them because there's some dependencies there. So it's a challenging space in which the task is usually one of integration. And so it's working with your existing systems, finding where you're missing a system, and then adding another system to your whole collection in order to solve that. And it's a terrible way to do business. But that's the state of the industry right now. It's a huge challenge. Again, just trying to do change management. Like I sat through with another pharma company, there's a long process of picking a new LIMS system. And they had consultants from all over the place coming in, but every department had different needs and different things. And so just the challenge of getting buy-in from everybody to make a change like that is difficult. What we've found, it's obviously much easier with small biotechs or startups that really, they're just using Excel right now, and you can move through that. And so we can do everything from helping to find existing tools to developing custom tools for a certain area, or being able to just help sort of integrate what you have and find a way that you can start charting out that path to a better informatics ecosystem within your organization. But it's not an easy task.
All right, thank you very much.
Thank you.